The next presentation, Like Father, Like Son, is a collaboration between Dr. Yuri Dubrova and myself. Dr. Dubrova has provided me with a comprehensive high-level video presentation and many materials and references describing his research. My role has been to select and present the material that is best suited for this symposium. Dr. Dubrova is a professor of genetics at the University of Leicester in the UK. He received an honorary Bachelor of Science degree in biology from the University of Kyiv in Ukraine, and his PhD and Doctor of Science degrees from the Institute of General Genetics in Moscow, Russia. Dr. Dubrova is on the editorial board of the journal Mutation Research, Genetic Toxicology, and Environmental Mutagenesis. He is also a member of the Radiation Research Society in the US, the Environmental Mutagen Society in the US, the Association of Radiation Research in the UK, the European Mutagen Society, and the Committee on Medical Aspects of Radiation in the Environment. Dr. Dubrova's research interests focus on the analysis of germline mutation induction in humans and mice following exposure to ionizing radiation, chemical mutagens, and some anti-cancer drugs. His recent research has also involved transgenerational genomic instability in the offspring of exposed parents. It is my honor to now present the work of Dr. Dubrova. In collaboration with Dr. Dubrova, I will present research studies performed by Dr. Dubrova. The title of this presentation is Like Father, Like Son, Transgenerational Epigenetic Effects of Parental Exposure to Radiation. And Like Father, Like Son is from William Shakespeare. The first two studies that we're going to look at have been discussed in detail in the recent review by Yuri and Dr. Sarah Poltseva. This excellent review will be referenced many times during this presentation. The slide shows a little background generational terminology. Parents are called the F0 generation. Mating of the F0s yields the F1 offspring. Mating of an F1 with another F1 yields an F2. This slide is an overview of the experiments that we'll be talking about. Right at the top, we have the parents. And in these experiments, we're having irradiation to the male mouse. And as a result of that, we have mutation induction. Also, what's happening is that in the next generation, the next two generations, we see that we're going to have mutation induction in these two generations. And that this mutation induction of these non-exposed offsprings, this is what transgenerational instability is. This first experiment is the genetic effects of plutonium-239 salt injections in male mice. The mice were irradiated, and the number of lethal mutation controls the irradiated male parents and non-irradiated offspring were measured. And in this experiment, we saw an increase in the mutation rate in the irradiated parent and the offspring. This next experiment describes promotion of skin tumors by a potent carcinogen in two generations of descendants of male exposed mice to X-ray irradiation. The male mice were irradiated and then mated. The F1 and F2 were treated with TPA, which is a potent chemical carcinogen. The data shows that irradiation of the males before mating increases the susceptibility of progeny of at least two generations to increased tumor incidence. The data is shown on the right and this is from Vorobsova. This description of it is from Dr. Dubrova's review, but you could see this shows the increase in mutations for the F1 and F2. This is a very important finding. A question that came to my mind was, what if the F0 was a heavy smoker but not irradiated? Would treatment of the F1 and F2 with TPA have the same result? The previous two slides present examples of transgenerational effects following paternal exposure to ionizing radiation. There are several other examples in Dr. Dubrova's review. The next slides will present work of Dr. Dubrova. We've talked about this in the beginning, so now we're just going to look at the mutation rates. The thing that we're measuring here are the mutation rates at expanded simple tandem repeat sequences, and these are abbreviated ESTR, and we're going to be seeing that abbreviation throughout the presentation. These are short repeated sequences, such as GGCA, and then just a repetition of it. These sequences are in non-coding regions of the genome. There are no proteins or phenotypes associated with these sequences. And these regions were selective because they're sensitive to mutation induction. This slide shows the results. So what we see is that the mutations have been passed down to the offspring, as well as the exposed parent. This is quantitative results from that experiment. So we could see here the 5-fold increase to the parent, and then a 4.5-fold increase for the offspring. The next question is transgenerational instability mouse strain specific? And this is the design for the experiment. And right here, that's the exposed male, and these are the offspring, and these are in black. Control parents with no history of radiation are in white. For example, here and over here. So for the branch on the left, a male offspring is mated with a control female. The branch on this side is just the opposite. Female offspring is mated with a control male. This slide shows the results for that experiment. And we see an increase in the mutation incidence in F1 and F2 for all strains of mice. This slide shows data for studies done with germline and somatic tissues. So in this experiment, we can see that mutations are detected in both germline, which is sperm, and somatic tissue with both strains of mice. This slide is a summary of the results for slides 11 through 15. Detection of mutations in nearly all of the F1 and F2 offspring indicates that classical Mendelian-type genetic mechanisms are not operative. This suggests that the mechanisms responsible for the mutation are some type of epigenetic mechanism, meaning the mechanism for initiation of the effect does not involve a change in DNA sequence. The effect is genome-wide, as seen by the fact that it is observed in germline and somatic tissue, and is present in both F1, male and females, and F2, male and female generations. This slide shows the incidence of DNA single-strand and double-strand breaks in the offspring of the exposed mice. The picture on the right here just illustrates a double-strand break. And what we're looking at on the left are the mutations relative to the controls. So here we can see we have an increase in the single-strand breaks for both strains of mice, and double-strand breaks, an increase in those for both strands of mice. Unrepaired and uncorrected double-strand breaks and single-strand breaks are highly mutagenic. These provide a plausible explanation for elevated predisposition to cancer among the offspring of irradiated parents. Transgenerational instability is attributed to the presence of a persistent subset of endogenous lesions, and these are the types. This slide shows the frequency of HPRT mutations in the spleen of offspring. And we can see here, these are the results for the irradiated, and here's for the non-irradiated, and we see the same effect with both strains of mice. So the key here is that mutations are found in protein, and in this case, the protein is HPRT coding region of a gene. And this is in contrast to the previous experiments where we're looking at a non-coding sequence. This slide shows the effects of ionizing radiation, a strong mutagen, and 3 anti-cancer drugs. We see an increase in mutations following treatment of male mice with mutagens or anti-cancer drugs, as well as radiation. This is a very interesting slide right here. Here we're looking at the F1 offspring of irradiated male or irradiated female with unexposed mates. Note that the F1 offspring of irradiated females did not show any increase in the ESTR mutations. And that what we're looking at is right down here. And the results are the same for females irradiated as an adult, right here, or in utero. On the other hand, irradiation of the male mice resulted in the observance of ESTR mutations. The mechanism for these results is not known. The mutation rates are dose and dose rate dependent. A dose-dependent threshold was observed for the mutations. You can see that threshold right here. The levels of 0.1 guy and 0.25 guy are below the threshold, and these are in the range of those used for radiotherapy. On the other hand, at 0.5 guy, we have mutations. Note that with 1 guy, there are mutations with acute radiation, but for chronic irradiation, the result is small or negligible. These results suggest that most doses of human exposure to ionizing radiation, including radiotherapy, may be unlikely to result in transgenerational instability in the offspring of irradiated fathers. All right, another summary slide. Ionizing radiation can cause single-stranded and double-strand breaks in DNA of offspring. Mutations can be found in proteins coding sequences, as well as non-coding repeat regions of DNA of offspring. Treatment with mutagens and some anti-cancer drugs can also induce mutations in the ESTR regions of DNA. Irradiation of female mice, whether as adults or in utero, does not result in ESTR mutations in the F1 offspring. There's a dose-dependent threshold for the occurrence of ESTR mutations in the offspring of irradiated mice. Right, on this slide, we're getting into high technology with this and with the following study. This study is about the paternal irradiation perturbs the expression of circadian genes in offspring. And circadian refers to biological timing. This experiment quantitates and compares the level of mRNA expression for the offspring of irradiated versus non-irradiated male mice. So here we see a double-strand DNA, and when it goes through the transcription process, it produces messenger RNA. this is what we're going to quantitate. And then over on, this is for the irradiated mouse. On this side is for the non-irradiated mouse. And in this case, we're looking at the mRNA prepared in this setup. These are just example results for a single gene. So what we have here is we have the counts for irradiated and non-irradiated, and then we're looking at the gene expression status. So if there's no difference between the two conditions, we say that gene expression is unchanged. There's an increase in the irradiated relative to the control. We call this upregulation of the gene. If it is decreased, we call that a downregulation. For the actual experiments, the mRNAs for tens of thousands of different genes were analyzed simultaneously using what we call gene expression arrays. This is just a simple cartoon. And here we can see an array. In actuality, each array has 135,000 different reaction well. Each well contains a different portion of the human genome immobilized on its surface. And then labeled test sample and reference, in this case it's irradiated, are labeled with different fluorescent dyes. They're then applied to the array to get results. So the results indicate which genes have been turned on or turned off as a result of the test condition. So what happens is they get fluorescent reads, and in the case of yellow dot, there's no change to gene expression, and with green, that indicates an upregulated gene expression, and the red means that the gene is downregulated. So the results from these array experiments. Paternal irradiation does perturb the expression of genes in the offspring. The genes perturbed by the irradiation include those involved in DNA replication, recombination, and repair. Also cell division and cell death. These changes could explain observed transgenerational effects in transgenerational cancer. Okay, this is another high-tech experiment. And this looks at the genome-wide effects of ionizing radiation on mutation induction in the mammalian germline. This study used gene expression arrays and whole genome sequencing for the detection of different types of induced sequence variations. As in previous experiments, male mice were irradiated and then mated with unexposed females. This slide shows the different types of mutations that were found in the irradiated offspring. The first one we'll talk about is copy number. This shows a chromosome with a single copy of one gene. And as a result of irradiation, there can be additional copies of the gene introduced into the chromosome. This again is called copy number variation. The next one is a single base mutation. For example, a G goes to a C or to a T or something else. So it's just a single one, base mutation. Another one that was found was a deletion mutation. And here we have a few series of 3 base pairs that were removed from the sequence. We also have an insertion, and we have just the reversal. Here, some more bases were added to the sequence. This next one is very interesting. This is called the multi-site. And this mutation was found with the highest frequency in this experiment. And note the irradiation symbol right here. This is thought to be this type of mutation is thought to be a signature of radiation exposure. So in this type of mutation, we have more than one event that's occurring along a single turn, or it could be within 1 to 2 turns of the base of the DNA helix. This next slide is from a recent review, and we're talking about now the mechanisms for this phenomena that we've been seeing. At the top, is shown different environmental stresses. Note that radiation is one of them. There are others. Here's solar radiation, diet, stress, exercise, osmotic stress. And what we're seeing then is an unknown signal that's transmitted to body tissue and to germline. And it's then, there's a message translated to the F1 and other generations. The potential mechanisms for this that have been considered include DNA methylation, histone modification, and non-coding RNAs and prions. So these mechanisms could regulate how this non-genetic information could be transmitted. We're going to talk next about these non-coding RNAs. An example of these is called a microRNA, and it is the small single-stranded non-coding RNA molecule. This picture shows the central dogma. So we have our double-stranded DNA. It gets transcribed to give messenger RNA, and then it can be translated to give the polypeptide, which is the protein. The microRNAs can bind to the messenger RNA, blocking this process. This is an epigenetic mechanism since there's no change in sequence involved. In Dr. Dubrova's review, he has a detailed discussion of this effect. This is a schematic that explains the effect. Here we have the mouse, and it's irradiated. As a result of this irradiation, these small RNAs, this is a microRNA as well, this is in there, they're packed into extracellular vesicles. And that's shown right here. These vesicles then can cross the cell membrane, get into the blood vessel. From there, they get to the testis. From there, they get to the sperm. And from here, they can get to the offspring. So this describes a potential mechanism for small non-coding RNAs as mediators of transgenerational signals. These are conclusions for Yuri's review that is referenced down here. The transgenerational effects of paternal irradiation on the fitness of their offspring in mammals still remains poorly understood. The key question is whether transgenerational effects can be observed in populations inhabiting radioactive contaminated areas. Organisms inhabiting radioactive contaminated areas are continuously exposed to ionizing radiation. Therefore, the analysis of transgenerational effects is complicated. Mutational studies of the offspring of irradiated may not distinguish then between the direct effects of exposure, mutation induction, and transgenerational effects. We have analyzed the effect of chronic low LET gamma irradiation on the manifestation of transgenerational instability in mice. However, it remains unknown whether paternal low dose or chronic exposure to high LET sources, such as alpha particles and fission neutrons, can significantly compromise the offspring of irradiated parents. Plutonium-239, the major source of ionization contamination at Rocky Flats, is an alpha emitter. Yuri's conclusions for the presentation. High-dose acute paternal exposure to a number of mutagens can significantly destabilize the genomes of their offspring. Transgenerational instability is a genome-wide phenomenon which affects the frequencies of chromosome aberrations and gene mutations. Transgenerational instability is triggered in the directly exposed germ cells by a stress-like response to generalized DNA damage. Transgenerational instability is attributed to the presence of a persistent subset of endogenous DNA lesions. Transgenerational instability is attributed to the epigenetic changes affecting the expression of a subset of genes involved in rhythmic process and regulation of transcription. Transgenerational instability may represent an important component of the long-term genetic risk of human exposure to mutagens, but we need human data to prove it. These are Yuri's acknowledgements. As you can see, there's been a lot of people involved in this, and this work has expanded over 20 years. I would like to thank Dr. Dubrova for the opportunity to collaborate with him for this presentation, and also Fran Levine, an occupational therapist, and she's on the PSR Colorado Board of Directors, for her careful review and suggestions for this presentation.